Okay, so we will start. Good morning, everyone, and welcome to another Bureau Friday seminar series. Today, we are very delighted to have a speaker from Norway, Dr. Nicole Dovbell. She studied biology in the University of Tübingen. Did I pronounce it
Yeah,
correctly?
yeah, very good.
Tübingen, Germany, and obtained her PhD in geomicrobiology. From 2014, she worked as a lab leader in the company BASF on oil field and industrial related microbial problems. Since 2019, she works as a senior researcher in the Norway Research Institute NORS, Norway, with the main research areas in the microbial effects during hydrogen underground storage and geothermal installations and biogas production. So we are very delighted to have you, Dr. Nicole. The stage is all yours now.
Thank you so much for the introduction, and thank you again for inviting me today. I always enjoy to show this topic, especially to a very interdisciplinary audience. So yeah, let's get started about microbial side effects of underground hydrogen storage. So first, because I know that probably not so many microbiologists will be present in the seminar, I thought I'd start very basic with the question, where do we actually find microbial life, and why I'm actually presenting here for a topic on hydrogen storage. So a simple answer I was given is you can find microbial life almost everywhere. So you can find them in soil, in sediment, in solid rock, of course, in water, on your skin, in your mouth, in the guts of from all the animals, including us, in hot springs, and also oil and gas reservoirs. So when I talk about microbes or microorganisms, I want to first define a little bit what I mean with that. So there are actually three major groups of life known to us. You can see a very simplified tree of life here, which was published in 1990. It was a really big deal. So you can see that we actually cluster the types into bacteria. I think everyone heard of them. And then the archaea, and then the eukaryotes. We are part of the eukaryotes, you can see here. That's the animals, the fungi, the plants, and a few other things. So we are actually coming out of the line of the archaea. So archaea are not so well known as the bacteria, but they are very important for the subsurface. When you now look under the microscope, for example, for bacteria, you will see single cellular organisms in a micrometer range. And now you wonder, how about archaea? That's the same. You will also see singular small cells. But then again, eukaryotes like yeast are also small singular cells. So there it will be a little bit tough to distinguish them. So in my talk now, I will focus on the subsurface relevant ones, and that's the bacteria and the archaea. And as I said, this tree of life is actually very simplified. Now looking at a more recent one actually shows you already the abundance and diversity of these groups. So you can see this is now based on DNA sequences all around the world, that this is the big cluster of bacteria. Here we have the archaea, and then the small thing here down, that's actually us, the eukaryotes. So you can see that when we talk about bacteria, the diversity of metabolisms and groups is enormous. So now going a little bit into detail. So the difference between a bacteria and archaea, I will not go into detail because that's too far. As I said, they are unicellular organisms, similar size, and they can grow on a variety of energy sources. They have some very specific differences in the cell walls. But for now the hydrogen storage aspect, the most important difference is that bacteria, which can have like normal shape of spheres and rods and filaments, when they see CO2 and hydrogen, they will produce acetate. Archaea, on the other hand, their shape is very different. They can have every shape. Then if they see CO2 and hydrogen, they will form methane. So there are no bacteria known up to date which can actually produce methane. So, and just like when you now wonder shape everything, what does that mean for Archaea? It means really they can have all the shapes. Here's a very beautiful picture of a Archaea isolated from Egypt, which is called Halo quadratum because it's square. So if you see this under the microscope, you can already say, okay, this is an Archaea. So where do we find microbial life? So the Archaea, they tend to live a little bit more in the extreme environments like the hot springs, whereas bacteria, they rather like the more ambient conditions. And there are different factors now shaping microbial communities in the subsurface. The most important by far is the temperature. So there has been life detected on this planet from -20 to 122. As long as water is liquid, in its liquid phase, you can find life. Above 122, nothing has been found yet. So that means if, for example, your reservoirs are very, very hot, way above 122, you can be sure that there will be no microbial life. It will be sterile. Here I always like to add, from my experience from the oil and gas industry, please always be aware if somebody says, yeah, we have a really hot oil field, 120 degrees easy, then always ask for current downhole temperature measurements. Because when they discover the reservoirs like 40 years ago, maybe it was 120 degrees. But if they then inject seawater or something for decades, around the injection area, it will not be 120 degrees anymore. And then there could be quite a nice temperature for life there. So always be careful here. So another factor is pH. So the most diversity you will find at neutral pH, but there are specialists in all the extreme ranges. Salinity, absolutely no problem. Again, specialists you can find up to saturation. Same with metal concentrations, can be like toxic heavy metals, can be a factor. Pressure is an interesting one. There are actually no pressure limits identified up to this point. Permeability is a aspect, especially for reservoirs. Give me a second. So I had to get my cat out of the room. So permeability, if you have a really, really, really tight reservoir going into the millidarcy range, then you actually don't have enough physical space for microbes to exist. So this is also a point here. And then of course, nutrients. What kind of nutrients are present? What kind of carbon sources? Is there phosphorus? Is there nitrogen? All these factors will shape your community, and all these factors will also change what you will observe in the subsurface. So when we are now saying, okay, there's a lot of microbial diversity in the subsurface, what are they actually doing? So it depends what is there, but then again, what is possible? So we start with normally organic matter input, which is broken down into more simple components like sugars, lipids, and amino acids. They can be fermented by microbes into even more simple substances like acetate, hydrogen, and CO2. So, and then the so-called redox cascade starts for microbes. So first, what will happen is microbes will use oxygen to actually use these products. Oxygen has a lot of energy. That's also the reason why we use it. So they will grow first. When oxygen is then depleted, which is actually happening very, very fast, you come into the anoxic zone, which is actually most of the subsurface. And then first there will be nitrate reduction, then there will be manganese reduction, iron reduction, and at the end there will come the nanosulfide reduction and methane formation. So these metabolisms actually all compete for the same carbon sources and also electron donors. But this explains, for example, you may have heard of that oil and gas very often inject nitrate. The reason is when you have nitrate present, nitrate reducing microorganisms will outcompete sulfide reduction. That means that you will inhibit sulfide reducers and therefore will not have H2S. And yeah, so you can already see here, you can see the hydrogen, so we're closing into the topic. This is, of course, again, very simplified. You can spend your whole life just studying one of these metabolisms, and people do that. So here in this slide, I just want to show you how one of these so-called cycles actually look like the sulfur cycle. So this is just now the microbial reactions going on in the environment. So it's crazily complicated depending where you are, just cycling between sulfate and H2S. So as I said, hydrogen, this is actually something microbes use. You may wonder, and I can say, yes, hydrogen is very different to methane. I think a lot of people know that it's very different from the physical aspects like density, the heating value, and so on. But methane actually cannot be used by microbes very well, in very stark contrast to hydrogen. Hydrogen is a universal electron donor for microbes. That means that hydrogen actually can fuel all these metabolisms I just showed you. That means the sulfide reducers can use hydrogen, nitrite reducers, iron reducers, even there are microbes using oxygen and hydrogen. So hydrogen is actually was named the oldest electron donor for microbes on this planet because it has been around for so long. So it's actually perfect food. It's perfect food for microbes. So when you now think with this precious hydrogen and you want to store it in the underground, that this will be in direct contact between a huge diversity of microbial life, which can possibly feed on hydrogen. So this is actually one of the big questions: is what will happen? Will there be hydrogen loss? What types of metabolisms will be active? Maybe there will be multiple processes at the same time. So this is a topic which has not been researched very well, and it's now just recently starting. So how does that actually microbial hydrogen consumption look like? So this is now from one of my projects, which I'm doing together with the University of Bergen. It's the HYPE project, where we are looking at different metabolic groups and their potential hydrogen consumption. So what I'm doing here is I'm culturing microbes in little glass bottles. You can see here one of them. I'm providing everything they want. This is now a sulfide reducer called Olido desulfovibrio alaskensis. And then you can see I added different amounts of hydrogen in the headspace, 10%, 40%, and 90% of the headspace of the gas phase was hydrogen. And as you can see over time, the hydrogen goes down and is eaten up completely. Then you can get maximum rates, all the nice kinetic data for this specific strain at this specific conditions. So every strain, every microbe will be different. They have different growth rates, different speed of growth. It also depends if they grow on their optimum temperature or maybe a little bit lower and so on. So this is how it looks in an experiment. So now coming to the hydrogen underground part. So with now hydrogen being talked about like one of the energy carriers of the fossil-free future, we need to store a lot of hydrogen. We can't just store it in tanks. That's just not viable. We need much more volumes. So the idea is actually to use maybe depleted gas reservoirs, salt caverns, or aquifers for hydrogen storage. There you can store enormous volumes of this gas, which you can withdraw out of the subsurface when you need it. For example, the idea would be you produce a lot of hydrogen during the summer when you have an energy surplus, and then during winter you withdraw it out of your storage site when you need a lot of energy for heating your homes, and then you can use the hydrogen there. Here, this is now, this picture is taken out of my review paper I published in 2021. So there you will find much more details. So if you're interested in this topic, I advise have a look in the review paper. Because every topic like depleted gas reservoirs, salt caverns, and aquifers, it's 3 very different topics because we're talking about different conditions every time. So in the following slides, I just want to touch now upon a few of the most relevant studies which has been done on this. Yeah, so for the depleted gas reservoirs, just to get everyone on the same page, the temperature and salinity range are enormous. You find everything from 20 degrees to whatever temperature, salinity same. And the experience for hydrogen storage is not very high, you can say that. Artificial salt caverns, they have a temperature range maybe between 20, 60, sometimes up to 80. Of course, super high salinity. And here we have at least experience from hydrogen storage, at least in 3 caverns in the world. With deep aquifers, there has been absolutely no experience. And then again, deep aquifers have a very high range of temperature and salinity. So everything needs to be assessed separately. As mentioned, there have been field trials for hydrogen underground storage, or not even field trials, but ongoing storage sites. So here I want just to mention it's 4 salt caverns. There's 1 in the UK, in Teesside, and 3 in the US. Actually, I think they're all in Texas. And they are all under industry leaded. It's for the chemical industry. So they are using the salt cavern to store hydrogen more on a daily basis to feed into their chemical plants. Unfortunately, there is absolutely no data available on these kind of salt caverns. Many people have tried to get access to this data, but they are not willing to talk to us. So if this is going really, really well, or maybe not, nobody knows. At least, I mean, they've been in operation for quite some time. So then there was a field trial actually in a saline aquifer in France, and an aquifer in the Czech Republic, and then also 2 field trials in depleted gas reservoirs. And here, actually, in every one of the porous media storages in both aquifers and the gas reservoirs, microbial activity was clearly shown. and hydrogen consumption was observed. So this is not something which seems to be like a problem for maybe some local sites. This is really a major problem which needs to be assessed. So going a little bit more into detail in the porous media storage, because this is something which has for sure the highest volumes. So what we know from aquifers and hydrocarbon reservoirs is that they, if they are in the right temperature range, contain a very diverse community. They are either already like indigenous there, or they have been introduced by operations. Sometimes it's very hard to distinguish. And we also know that the communities differ in every single reservoir, because every reservoir has different conditions. As I said, like different salinities, different temperatures, different water chemistry. That means you will find different communities. The porous structures are actually aimed for really long storage cycles, because it is more, you can store higher volumes, but it is more energy expensive to actually inject these high amounts of hydrogen. So there you actually we're talking about seasonal storage. But if you store these hydrogen for so long time in your porous media, you have a lot of time for microbial adaptations and activity. And you have a very large surface area. Surface area is in this case really bad, because if you have a large surface area between gas and your brine, which contains the microbes, you will have more activity. So what could happen? You can lose your precious hydrogen. That's one thing, because they just eat it up. You could produce H2S, which is, I guess, the worst case if you then inject your precious hydrogen and you get H2S out. You could also get methane. I mean, that is not as bad, but for sure not what we want. You can get severe corrosion from the microbes. There are a lot of microbes which can corrode. It's so-called microbial influence corrosion. It's a very big problem in the industry. Like microbes can eat through steel pipelines. So they can actually be triggered. You can have reactions with minerals. And if you have very intense biomass production, you can have plugging of the pores. So now recently I think a week ago, there was now a paper published from the German BGR, where they looked at fluid from a gas reservoir. They incubated it over, several days with and without pressure. And it's relatively easy to understand what we're seeing here. You have your hydrogen in the headspace, and whoop, they eat it up. They refeed the hydrogen. It goes down. They refeed the hydrogen, and they consume it. They consume until they actually run out of sulfate. So they got an intense H2S production and a very, very long hydrogen consumption. So this was now at 30 degrees for a field in Germany. So this already shows this is really a problem which can be really major, especially in the beginning of your storage. Then I want to show again some data from our HYPE project. This is now a paper from my very good colleague Na Liu. We hope to publish this paper very soon. So what we're doing here is actually microfluidics. So microfluidics, you have chips where we actually printed a porous media structure. This is the black area you can see here. And then we can fill this porous media structure with brine, and we can also inject hydrogen. So the light gray is actually the hydrogen. So what we did, we used microbe, again, a sulfide reducer. We injected it together with the brine into the model. And then we injected the hydrogen and closed everything to mimic a storage. And I think you can see it quite well when you focus on the gray part, which is the gas, the hydrogen gas, that the bubbles move and they get smaller and smaller. They disconnect, and we actually see a loss of almost 70% of the hydrogen after just 1 week. So this is, of course, has several problems, not only that you get the loss, but also because of the disconnected hydrogen bubbles, you will have quite a tough time to reproduce your hydrogen out of the porous space. If you look a little bit more into detail, you also can see again in dark black, that is the solid. Then we have here the hydrogen, and this is the brine containing the microbes. So in the beginning, you can see here the contact angle of 24 degrees. So that is what we expect. And already after 1 day of microbial activity, you see that the hydrogen reduced and moved, and that the contact angle changed significantly. And what we also see, which we only see with microbes, is that sometimes it's really like these very straight lines of the interface between the gas and the brine, which looks super weird. But I think it's also because the microbes, of course, they need the hydrogen, so they will actually tend to stick to the gas-brine interface. And this is also the reason why they will change the contact angle. So they have been known to do that, many microbes. So this is also a fact which needs to be considered if you are planning your hydrogen storage. And we also hope that these data will be funneled into new models, because right now, when you look for modeling of hydrogen underground storage, microbial effects have not been taken into account at all. So a very famous example for a field trial in porous media is the Underground Sun project, which was conducted in Austria already several years ago. And also there they found microbial consumption and conversion, both in field tests, in the field test, and also in the lab tests they had before. So you can find everything in their end report, which is available online on their website. So you can see in blue, the hydrogen goes down over time. And in this case, they actually get methane produced. So this time they don't have a problem with sulfide reducers, they have problems with methanogens. And this is now actually what they published also for their field trial. So in blue, that is what they injected, hydrogen into their storage. And then you can see what they actually reproduced. And at the end, they lost around 3% of their hydrogen in their first storage cycle, which sounds now maybe not that bad, but again, they only injected 10%, and then losing 3 is quite a lot. The storage cycle was also not that long, and very interesting would be now to see if they would now inject hydrogen again, if the loss would be even more intense. Because you can think, if now the microbes actually know, oh, there's hydrogen is coming, now they can be active. But for sure, there are much more field trials needed on this topic. Field trials with very good monitoring schemes. Then I want to touch a little bit on the salt caverns. As I said, salt cavern storage is a thing already. They are artificially leached into salt structures all over the world. And they are not sterile. So you will get actually cell input from either the leaching process or in the salt itself contains already microbes. The conditions are extreme, but this is not a problem for the specialists. So you will find microbes. The storage cycles are expected to shorter, so days to weeks, which reduces the overall consumption. But then again, if you have regular input of fresh hydrogen, this is also nice for microbes. And the temperature and pressure is well within microbial limits, so there's absolutely nothing to plan there. So what could happen? Again, you could have hydrogen loss. You can have H2S formation. Methane formation is probably not a thing in salt caverns, as far as we know. Then again, you can have corrosion and reactions with your minerals or your salt structure itself. So this is something also which has not been studied well. So this is now data from a paper also published. This is a DNA community plot. So every single color you see in this plot is a different microbial family. So they looked at a lot of different salt caverns. They are all located, I think, in the eastern part of Germany. And in each cavern, they found microbes. And in each cavern, actually, the community looked quite different. So this is for a microbiologist like me, this is like, oh, this is amazing. Each salt cavern is its own little universe, which needs to be researched and explored. It's amazing. Of course, for storage operators, this is like worst case. That means every single cavern needs to be assessed separately for the microbial risk. So yeah, there are many open research questions, as I hope I could convey that to you. So we don't know which microbial types will be crucial. We only can speculate right now. It will be the sulfite reducers and the methanogens, but there are others. Then again, are they always the same? As I said, maybe every cavern a different universe. But maybe also with hydrogen, maybe always the same key players grow up. We don't know yet. We don't know about the microbial rates, which are very important for the modeling. There are no reported studies for the pressure effects, or maybe some kind of thresholds. We can estimate it on literature data, but this needs to be always taken with a grain of salt, because we can only estimate what we know. And these salt caverns, as I said, they have not been investigated very well. Is inhibition possible? That is something we need to look out. I have very good feeling that within salt caverns, we can get this under control, because it is more or less a confined space. In a porous media, that will be tough. We know that already from oil and gas, where they have souring problems. If you have an active field producing H2S, it will be super difficult to get control of it. Then a very big topic, geochemical effects together with microbes. Not studied at all, but very important. So everything can actually influence each other. And yeah, there's also the idea, maybe we could use these microbes as a leakage detection. When you actually look for specific types in the surface, maybe you can use them as a marker. But this is just also an idea. Nobody worked on that yet. So at the end, I want to make a big shout out that we need cross-disciplinary projects. I know everybody likes to stay in their own field, but it is really, really important to talk to each other. So the microbial data is really important for the modeling, the safety, the material selection, the storage selection, and so on. So I'm always hoping that we have cooperation between the academia and the industry, between the sciences, so that we can actually combine the knowledge. And it is very important to keep microbes in your mind, because this activity, which can happen in your storage site, could actually be the showstopper for hydrogen underground storage. So it is the identified main risk right now. So we need to understand it and study it more in detail. And with that, I'm finished, and thank you very much. And in case there are any questions, I'm happy to answer them.
Thank you, Dr. Nicole. That was a very interesting talk, and we learned definitely. I will ask our listeners, you can mute and ask the question or write it in the chat box. I see one question in the chat box. What is the source of sulfur in microfluidic experiments?
So in the microfluidic experiments, we did the source of sulfate. It was an artificially made media for the microbe. So it is this microbe has its media, which you can just read up in the literature, and you just add sulfate, mix it in water with some salts, and then it grows on it. So it was specifically for this microbe. Of course, like in the environments, it's different. Like in porous media storage, you have sulfate input from the sulfidic minerals or from seawater. And in salt caverns, you have also sulfidic minerals, specifically anhydrite.
Thank you. And maybe I add a second question. That was my question. Do we know that these microbial communities in the subsurface, these are naturally there, or some of them are contaminations that are coming from the surface?
Yeah, that's a very good question. It is so hard to tell because sometimes there are no recorded data, like from the initial part, like what was your reservoir initially and what was it afterwards. I think it will be always a combination of both. So that you will always have contaminants from your operations. It's just a question if they will be then the dominant ones in the subsurface, but they will be there.
So there's another question by Helen Nee. What methods can be used to abate microbial activity in porous media in the subsurface?
Abate? What does that mean, this word? I'm sorry. I don't know this English word. What does abate mean?
Do you want to unmute and ask the question, Haloon?
Sure. I just mean to reduce the activity.
Ah, thank you.
No problem.
Sorry.
No problem.
This
Thank you.
That's a very specific word. Okay. Yeah, to reduce the microbial activity in the subsurface in the porous media. Yeah, so for one, it's always good to reduce your input of organics. So reduce your input of carbon, reduce your input of sulfate. So this is, for some example, many reservoirs try to reduce sulfate when they're using seawater. So they have actually massive installations to reduce the sulfate. Yeah, that's actually more or less it. I mean, you can also add biocides to your injection water. This is, of course, also something which is done regularly. And I think if you design it in a very good way, especially in the beginning when you start your storage and so on, biocide treatments can actually help. But you need to monitor it also very well. So that's the things. It's really other ways to control activity in the subsurface is very, very difficult.
Okay, so we have another question by Phil Bennett. Thank you for the excellent presentation. And he's asking, do you have any other information on salt-tolerant hydrogenotrophic methanogens, such as methanosarcinaceae?
Ah, there is a microbiologist present. Yeah.
Sorry if I didn't pronounce.
No problem. Yeah. These are like the crazy names of microbes. Yeah. Yeah, that's methanosarcinaceae. So the interesting thing about these hydrogenotrophic methanogens is they seem to be very limited in their salt tolerance. At least that's what we know so far. That is because they live on their energetic edge. Because growing on hydrogen in CO2 gives you not a lot of energy. It's a very tedious metabolism. But living in salt requires a lot of energy for the cells. So there seems to be a cut that they, at some salinity, these microbes tend not to be able to gain enough energy through hydrogen consumption. This is the reason why I also said, like, methane producers and salt caverns are probably not a thing, because until now, nobody ever found them over, I think, 12% salinity or something. It is an energetic constraint. But then again, maybe we just have not found them yet. That is always to be kept in mind.
There is another question by Helen Nee. Are there any microbes consuming CO2?
Oh yeah, plenty. Many, many, many. Not only the ones growing on hydrogen and CO2, as I said, forming methane. CO2 is a very, very good carbon source for many microbes. They can fix the CO2 and use it as directly as a carbon source. That is, I think, for many subsurface microbes, where CO2 or carbonate are the only available carbon sources they have. So that is very widespread.
Mm-hmm. There's another question by Mark Schuster. Great talk. Have you or others investigated microbial communities in naturally occurring hydrogen systems?
Oh, a very good question. No, I have not. Nobody did, as far as I know. I'm not aware of anyone. There are like a handful of natural hydrogen seeps. For example, in Australia, there you can find them. I think they called like witching rings or something. They're very interesting surface. Like they form things on the surface, which looks quite nice. And I think there's one or several in Africa. No one really looked at the microbial communities there. They have not even been studied very well on the geology or the geochemistry yet. So, and there are more geologists and geochemists interested in this topic, and even they have not come to it. I was recently at a conference in Perth, and they were actually also talking about this topic, to go there and maybe take some samples. For sure, it would be super interesting to do that.
Thank you.
So I had a simple question. I'm just wondering where the bacteria living in the salt are getting their water from.
In the salt cavern, yeah, the salt caverns, they are actually never completely dry. So when you leach a salt cavern, they leach it with water, either with seawater or with river water or pond water, whatever is available. And so this leaching process will take quite a long time, up to a year. And then when they actually withdraw the liquid, they will never be able to withdraw all the liquid. There will always be a sump. So it's called a cavern sump, which is around 10% of the cavern volume. So there you have your leftover water, extreme high salinity, of course, saturated. And there you have all the non-dissolvable minerals also left of the leaching process. So there will be always this remaining liquid. And that also means that you have always a liquid phase on the cavern walls. There's some very interesting thermodynamic modeling that they actually say when you have then hydrogen injection withdrawal with the pressure changes, that you actually get evaporation of the cavern sump and it will rain in the cavern again. So it's a very nice paper on that. Not sure if it happens, but at least it was estimated.
Okay, so that water is human, is introduced by humans.
Yeah.
Those microbes are living in that salt before that cavern is artificially leached, right?
Yeah. Yeah. So there
must be some water in there, natural, at some very low level. Okay.
Yeah. So when you look at these salts, you will always find water inclusions in these salts. They can be really, really small water bubbles or inclusions, and actually relatively large. You can see it with your eye. And these water inclusions, they either come like geologically, but it's also been shown that these water inclusions, they travel through the salt. So and within these inclusions, they have been microbes also isolated.
Okay, thank you.
Hi, I really like a talk. I have two questions here. The first one is about your metabolic hydrogen consumption in your laboratory conditions here, because we know that there are two processes here. One is the hydrogen dissolution in the water, and then it's biologically reactions with molecular hydrogen here. So when you measure the hydrogen consumed, so first question I want to ask is how you emulate the effect of hydrogen dissolution in your laboratory conditions, because if this process is slow, and that means your measured the rate could be also slow. This is my first question. My second one is, we know that this hydrogen is stored in the subsurface. There are many different reactions, like the reactions with microbes and with minerals. Also, there's hydrogen exchange with formation water, like in the Mali reservoir there. So my second question is, how can you identify the microbial activities among these different reactions there? Yeah. Thank you.
Yeah, no, good question. So the hydrogen, first question, the hydrogen dissolution. In hydrogen dissolves super badly in liquids. So especially if you increase the salinity, then the hydrogen solution is really, really low. And you can calculate it. And what we calculated was that when you get contact hydrogen liquid, that the dissolution will be actually what can dissolve will be relatively fast. So you have actually reached equilibrium maybe in half an hour. And the microbes will be, of course, they need much more time. So they need days and weeks. So that is how we say, okay, everything which is directly in the beginning, the first few hours, this is dissolution chemically happening, and then afterwards that is the microbial activity. What we also saw, if you dissolve more hydrogen in the liquid, for example, by increasing the partial pressure of hydrogen, or have active mixing, to really get hydrogen dissolved, the microbes will really like that. You will get increased rates because microbes, they only can use the hydrogen which is dissolved. They cannot access the gaseous one. So the more hydrogen you dissolve in a liquid, the better for the microbes.
So you mean that in your labs, you want to try to, increase the contacting surface between the molecular hydrogen and your something kind of gas and liquid, something like this. In that case, more hydrogen can be dissolved in this formation water. Yeah.
Mm.
Okay. Thank you.
Yeah. Yeah. And your second question, it is always tough to distinguish geochemical reactions from microbial reactions because they're also interconnected. They can affect each other. So, for example, it's like if you get H2S formation, this is always a good indication that you have microbes, because microbes tend to form H2S. You can have, of course, also geochemical sources of H2S. So it's always good to monitor and measure as much as possible. So get all the data you can on the water chemistry, get all the data you can on the microbiology, the cell numbers, the community. Does it change over time? Get the information on the dissolved carbon in the water. Is it reduced? How is the pH changing? This all can give you then indications if it's microbial or geochemical. But sometimes it's maybe also both. It's difficult to say. It can help if you then do lab experiments to try to distinguish. But yeah, it can be sometimes quite tough to really pinpoint it.
Okay, thank you.
Hi, Nicole. Very nice talk. So I have a question about your Poroscale experiments. It was very, interesting that you observe, contact angle changes during, the microbial reactions. So I wonder if you ever tried geochemical reactions in your experiments. Do you think, these wettability changes happens because of geochemical, reactions in the system? Because to me, it's like that there is some mineralogy changes in the rock that you observe wettability changes. Or maybe there are some byproducts from the microbial reaction that deposit on the surface of the rock and changes the wettability. Do you have any idea why this happens?
Yeah, so in these microfluidic chips, everything is made out of silica glass. So if you would have now a complex rock, this would be even more complex. But I also, again, there I would expect wettability changes. We did a control experiment without microbes, just the media and the hydrogen, where we did not observe this. So it's for sure not a chemical reaction just from the media. So the microbes need to be there and active to do it. We have not pinpointed what the actual trigger mechanism is. So it could be several things. It could be a metabolite the microbes form, which is active, surface active. It can be the microbes themselves. This is an effect very well known as the so-called Pickering effect, because microbes, they have a cell membrane, which is, it's like fatty acids, and they are also surface active. So if you have a lot of microbial cells on an interfacial area, you will have interfacial and surface effects just by the cells themselves. So yeah, it could be both, and we have not found out what was now here the specifics. We just observed the effect for the first time.
All right, thank you.
Syed, do you want to go next?
Yes, I just want, maybe my last question at least. I guess Nicole mentioned that, CO2 is much more, better substrate for microorganisms. And I'm just curious, that we have done quite a few CCS pilots, right, in aquifers and sandstones, and there has not been a major report or issue of having these kind of, microbial reactions. Or so what is your comment on that as far as, translating those lessons to the hydrogen storage in the sandstones specifically?
Yeah, that's a good one. So with CO2 storage, you have to see that just pure CO2, it is a carbon source for microbes. But you need, of course, something else. So just CO2 will not trigger microbial activity. You need, for example, sulfate or something else. So this depends, again, very much on your aquifer conditions. And then again, supercritical CO2, which you tend to have during CO2 storage, especially in the beginning, during your injection. In your injection well, supercritical CO2 is very toxic. It's actually a disinfectant, which the food industry uses. So you can disinfect very good with supercritical CO2. So you will probably have a very strong toxic effect in the beginning. But I would assume now that in maybe deeper into your aquifer, where your CO2 then gets dissolved, and maybe in lower concentrations, where the pH effect is also not that strong, that you might get CO2 consumption. And there was a paper came out last year in Nature, where they actually observed it was in the US also, it was in US field trial. They observed that they actually got methane out of a CO2 storage site because they observed that microbes used the CO2 and they used the oil, which was still present. Was it an aquifer? No, it was a gas reservoir then, an oil reservoir. The oil, there came a little bit of hydrogen out of this oil, and then microbes transformed it to methane. So that was actually the first time that this was observed for a CO2 storage trial. So what long answer, but I can say the microbial investigations for CO2 storage are more or less not existing. So everything is a little bit more like we estimate. It can be maybe a problem in some sites, maybe also more on a long term, I would say.
Okay. Thank you.
If you have more time for one more question, to get back to the mitigation aspect of preventing microbial reactions in the porous media storage, don't you think that injection of a microbicide prior to the injection of hydrogen would perhaps take care of it?
It will ease the effect for sure.
Mm-hmm.
You will have a biocidal effect, especially around the injection well area. It depends a little bit then what will happen deeper into your storage site. Because from my experience, it doesn't matter how much biocide you will inject into a porous media, you will never disinfect it completely. The area, the volumes are just too big. There will always be an active exchange with the surroundings that you get new microbes coming in. So you will always need to retreat. So just injecting one biocide will help for sure, I think, depending on the biocide. So if it's not absorbing or something, but you have to monitor it very, very well to keep to be on top of it. But that is also something we're preaching to the oil and gas industry for decades. Like, please monitor your reservoirs for microbial activity, not just inject a biocide and then go away. So they're not listening to us. So hopefully someone else will.
So this is definitely something that needs to be looked into, right? It's different from production because here we inject and produce, right? Compared to oil and gas where you primarily produce. So it's a little bit different, but sounds like this is something that needs not just the microbial side, but actually the flow behavior of the microbial side into the reservoir and then how it survives in there, right?
Yeah, exactly. So there will be, I guess there will be coming inhibition studies to see what kind of chemistry will be actually the best to use for this kind of storage. As you say, it is more like a huff and puff, like you inject, you withdraw out of the same well. So it needs to be adapted. So it's for sure something to investigate.
Thank you.
It's 10 o'clock. So before we wrap up, I just have one question from my side regarding the experimentation. You showed the experiment where you cultivated the bacteria and then in the gas phase, you injected hydrogen. Mm-hmm. So how long does it take to consume that hydrogen by the bacteria? And how do you measure that hydrogen is all consumed? I'm very curious to know, how do you measure that?
Yeah, so this microbial strain used up this like 30 milliliter of hydrogen in around 7 days.
Okay.
But this is, again, it's strain specific. If you use a different strain, it will take longer or it will be faster.
Okay.
And how I measure it, the important thing is you need to measure the pressure and the gas composition. So what I do, I have my bottles at a certain temperature. So I measure the pressure in the headspace.
Okay.
Then I measure the gas composition with a GC. And from that, you can calculate, because you have your volume in your bottles volume of gas, temperature, pressure, composition, and then you have the ideal gas law, and then you calculate it into moles of hydrogen, and then you get the line. It's really important to look always also at the pressure. Some people only measure gas composition, but this will not give you the whole picture because hydrogen, it has a very low density, it gives you a high pressure, and eating up the hydrogen, the first thing you will actually observe is a pressure decline. It can actually, they eat it up, pressure is reduced. They can actually, microbes, they can draw under pressure because they're just eating up so much. So this is something to for sure measure.
Okay. Yeah. And also one more question on your chips. Have you ever tried to use other chips than silicon one for your pore structure?
Not yet, no.
Do you think it will change if you have similar composition as what you see in rocks? There may be some side reactions which may occur.
Oh yeah, for sure. For sure, the complex minerals from like already have in course that will make everything different. The microbes will behave differently, that's for sure. They love to sit on minerals, not so much on silica.
Yeah.
You will have different wettabilities and so on. So
yeah.
that will make a huge difference, I'm sure.
Okay. Okay, thank you, Dr. Nicole. That was an amazing talk, and there were so many useful things which I have taken, and I think I'm pretty sure a lot of people have taken from your talk today. And thank you for arranging to our time zone and make it to the talk today. We are very fortunate to have you today. Yeah, thank you very much, and thank you all the listeners. We will be there next week again at 9 a.m. on Friday.
No problem. Then have a good Friday. Thank you very much.
Thank you. Bye-bye.
Bye-bye.